Well, so on to today's speaker. So Tim, thanks for joining us from Freiburg. So Tim Lammermann is currently a group leader at the Max Planck Institute of Immunobiology and Epigenetics that's located in Freiburg, Germany. He's been there since 2015. Prior to that, Tim had done his PhD with Michael Sixt at the Max Planck Institute of Biochemistry in Martinsgrad, Germany. And then he went to the US, where he worked with Ron Germain in the Laboratory of Systems Biology at the NIH, where he did some really beautiful work on neutrophil swarming. And today I think we're gonna hear about that work. And I think a lot of beautiful intravital microscopy and beautiful images. But before we get into that, Tim, something we like to do in these talks is ask the speakers sort of a question about some significant kind of part of their career that's helped them get to where they are today. So I'm curious if you could sort of share with us one of the most impactful decisions that's gotten you to where you are today. Oh, you're on mute.
Thanks so much also for hosting me today and for this invitation. And to your question, I think really one of the most impactful decisions was to work on neutrophils, which was a decision during the postdoc, which actually when I moved to the NIH and joined Ron's lab, the idea was a different one. And this was to work on macrophages and macrophage migration. And then I very naively just thought I had some preliminary data and everything sounded as if you just have to finish the project. But then it turned out these macrophages were so slow. And I got really afraid that with a 5-year J1 visa, this will never work. I will be stuck in this NIH basement and be unemployed after that. Yeah. But while doing these intravital images and microscopy, there were always these neutrophils moving around. So we reshaped the project a little bit. And this is how I started working on neutrophils, and which was a very good decision. And from that moment on, it went like this, and very fast. And I'm not unemployed. Yeah.
Yeah, and will you ever go back to macrophages? Macrophages are good to study too, now that you have a lifetime career.
That's true. So you will see that there are some, actually this project was finished now. But it required 7 more years and a PhD student. And I have some slides on that also in the presentation. Yeah.
Terrific. Okay, well, we look forward to hearing about your work, and if you want to take control of the screen. I just want to remind everybody that at the end of the presentation, Tim will join Twitter to answer any questions, and his Twitter handle is @Lammermann_lab, and you'll see that in the Global IMMUNOTALK Twitter too. Okay, so the stage is yours, Tim. Thanks again for joining us.
So again, also thanks a lot for this invitation, and I'm now really happy here to share with all of you really my fascination about the dynamics of innate immune cells and innate immune responses, and in particular the question, how do immune cells coordinate their dynamics in the complexity of an inflamed and infected tissues when these cells have to integrate so many different signals from different sources in situations like sterile inflammation, infection, and we also have some projects on anaphylactic tissues. And so the research questions that we have in our lab are: how do individual immune cells move and position in tissues? How do they communicate with each other and coordinate their effector functions? And also, how does this affect the outcome of an immune response? And what fascinates me so much, like having grown up as a more like as a cell biologist, is the coordination of these dynamics, because it includes events on the single cell level where each individual cell needs to do fundamental cell biological processes like directional sensing, cell migration, and cell adhesion. But then it also has like aspects on the population level, because cells are secreting attractants, inflammatory mediators, and cytokines, so they will communicate with each other, or some of them directly interact. And then you also have the tissue level, where the composition or the architecture of a tissue, or also parameters like diffusion properties in a tissue, also influence the dynamics of innate immune cells there. So what we try to do in the lab is to visualize the dynamics of these cells at multiple scales. So very often, most of our projects start with intravital microscopy, where we really directly look into the tissue and at the dynamics of the cells in the tissue. And then we move to what we call in vivo mimics, where we try to mimic what we see in the tissue in some maybe smart setups to visualize then single cell dynamics and cell-cell interactions. And then we also use some more classical imaging approaches to look at intracellular dynamics of moving immune cells. And with all that together, we try to get a comprehensive understanding of the dynamic behaviors during immune responses. So one of the cells we are focusing on are neutrophils, and in particular their dynamics in the interstitial spaces. So we all know neutrophils are key sentinel cells of the innate immune response, and they circulate in the bloodstream, and upon an inflammation or infection, they go out and move into the tissue. They start to explore the tissue and try to detect or sites of tissue damage, cell death, or maybe any kind of microbes. And when you give a talk about neutrophils, then you almost can't do it without showing this video here, where a neutrophil chases a bacterium in a blood smear. So this is a classic movie from David Rogers, and it's now 62 to 63 years old, but it already shows all the features of a neutrophil. Yeah? It shows this neutrophil, how it here kind of seems to smell the bacteria. It shows how the flexible cell shape changes that the neutrophil does to follow the bacteria. In the end, it will take up the microbe and then even digest it. So neutrophils are perfect sentinel and hunter cells. They are also equipped with several types of intracellular granules that contain antimicrobial proteins and proteases. And we also know that they have several antimicrobial mechanisms, including degranulation of toxic proteins, which are toxic for microbes, phagocytosis for the elimination of microbes. They can form neutrophil extracellular traps, and they can also die by forming nets in a process called netosis. So one of the most important and decisive processes for neutrophils is to get from the blood to a site of inflammation or infection. And this is in a process called extravasation, where the neutrophils undergo a multistep adhesion or activation cascade along the inflamed endothelium. So the neutrophils undergo sequential phases of interactions with the inflamed endothelium, which involves interactions between selectin ligands and selectins, chemokine receptors and chemokines, as well as integrin receptors and integrin ligands. And this then together causes these sub-steps that we know and that are then termed capturing, rolling, slow rolling, arrest, crawling, and then they move through the endothelial lining by diapedesis, and then they go out into the interstitial space. And we are not working on the process, but there are many colleagues and experts that work on this research area, which is still very active and has like still a lot of novel findings. And one of the things, for example, is that there are mechanistic variations regarding the substeps that I just mentioned, as well as the molecules involved for specific vascular beds when neutrophils move out in different organs. So our lab is more interested in what happens when the neutrophil has exited and went out of the blood and is in the interstitial space of a tissue, as you can see here in a time lapse over 30 minutes by recorded by 2-photon intravital microscopy. So in green, you see here neutrophils that are moving around and exploring the tissue. And it's safe to say that neutrophils are really very fast cells of the innate immune response. So they move fast in tissues. And with that, I mean, they can move up to 1 millimeter per an hour. And this makes them around 20 to 100 times faster than other myeloid cells, as for example, tissue-resident macrophages. What neutrophils also are is that they are also the prototype of amoeboid migrating cells. So neutrophils move in an amoeboid fashion, and this means that they deform their cytoskeleton, meaning they very quickly induce a front and back polarity, and they undergo rapid cycles of protrusion and contraction. This means that they can, for example, move through narrow pores in the tissue, like here through this constriction point. By squeezing, they can deform the nucleus, and this amoeboid movement is also adhesion independent. And this means now for neutrophils that it's very autonomous from their tissue environment. It's very flexible, so it means that it's not steered by some preformed extracellular matrix paths. So these cells rather follow paths of least resistance in the tissue, and this makes them very adaptive to many tissue geometries. And when you think about it, it also makes sense because a circulating neutrophil doesn't know in the end where it will end up. Is it the inflamed kidney or the infected macrophage, or the infected liver or the inflamed lung, or maybe somewhere in the brain. So it needs to adapt everywhere. Yeah? And by moving in an amoeboid fashion, it realizes this. So here I mentioned already that this amoeboid migration of neutrophils is adhesion independent, which is now kind of clear for most of us. But actually, when I did my PhD work together with Michael Six between 2004 and 2008, this was not clear at all. Yeah, because the prevailing cell migration paradigm at that time was that a moving cell makes a protrusion, it adheres to the underlying surface. That's the basis for traction, and then it de-adheres, and that's the principle of adhesion-mediated traction. And in the middle of all that are integrin receptors, heterodimers of the alpha and the beta subunit, which are the major mammalian adhesion receptors. And this is how it was thought that all cells are moving. And so during my PhD work with Michael, we asked, can migration without integrins in immune cells, is this possible? Yeah. And so we made a very complicated mouse. It's an inducible knockout with crosses with conditional as well as constitutive knockouts for integrin subunits. But this allowed us to generate from these mice primary immune cells where we genetically removed all integrins from the surface. And the trick was that we depleted key subunits, and with that we lost the whole heterodimers from the surface of these cells. And then we could study a number of different immune cell types. So the main cell at that time was dendritic cells, but I will also show you then the results for neutrophils. What we had as a second model was we also had a knockout for tailin-1. So tailin is a very important molecule to switch integrins from a low affinity to a high affinity conformation. And so with that, we could generate cells that were genetically impaired in their activation of all integrin family members. So when we then used these cells and just put them on a 2D dish, you could see the control cells, they just spread and then started to move around, while the integrin knockouts or the tailin knockouts, they of course just floated around. And if you're floating, you can also not move. Yeah. But everything then changed when we moved into a 3D gel, yeah, because now, as you can see here in this collagen gel, the cell has completely different options to interact with its environment. And what happened was that when we then put these dendritic cells that were completely naked of integrins on their surface, they just moved perfectly fine when they were moving up a gradient of CCL19. The same thing actually happened also when we looked at neutrophils. And here, these are just the control neutrophils that were chemotactically moved here in a 3D collagen gel. And on the upper right, you also see how the cell squeezes its nucleus and the whole cell body through the collagenous fibers. And also here, there was not really a difference between wild type and integrin knockouts. So since then, we have a different paradigm. And that's the one of 3D immune cell migration, and which is now considered that immune cells move in an amoeboid fashion and in an integrin independent fashion. And this has been shown for neutrophils, dendritic cells, also for T cells and for B cells. And very recently, we also looked into macrophages. So just in the introduction with Kate, we mentioned this project. So it took a while. So macrophages were interesting because they are notoriously adherent. So so if you put them somewhere on some kind of glass slide or so without even coating, they will stick there. Yeah. So we also put them in a 3D matrigel system. And when we had knockouts either here of TALEN1 or integrin beta 1, then these cells rounded up. They even did not spread in the tissue. And then we had to observe them over very long times to see their movement. Like these were migration experiments over 24 hours. These knockouts also did not really move. However, and this was the surprising thing, once we gave them chemotactic stimulus, again, they were completely fine and moved just like wild type cells and could perfectly move. So chemotactic macrophage migration did not depend on adhesive interactions. So this paper was just recently published and I can refer to it. I can't show you all the details, but what was really surprising, what we saw here, that macrophages have a surprising flexibility and contrasting integrin demands for either a chemotactic wound response migration, where they can move in an integrin independent fashion, or once they are in a random migration surveillance mode, where they move in an integrin dependent fashion. And this also had consequences, in particular when the macrophages do what they do, and this is removing things. And here they are removing apoptotic cells in the gel, which the wild type cells, which move and have show you this migration based surveillance mode, just clear within 20 hours all the dying cells. While these integrin knockouts, they can in principle, as you can see here, take up macrophages, but as they are missing this adhesive migration in the gel, they could not survey that well. And we also have some in vivo proof that this is also happening in the tissue. So just to sum that up here, not everything is told here about 3D migration of immune cells. So for macrophages, also once they move in a directional fashion, they can adopt this amoeboid migration mode, but if they walk in an undirectional manner, they require integrins, and we are still currently looking in other immune cell types and trying to get a complete view of and picture about amoeboid versus mesenchymal migration in immune cells. But now with this little detour about amoeboid immune cell types and macrophages, we now come back to our major topic and our amoeboid neutrophils. And already at the end of my PhD, and then also with the beginning of my postdoc, this is where I really became interested in neutrophils, and in particular because of two studies that were very remarkable. And these were intravital microscopy studies. The one was from Tatiana Chtanova and Ellen Roby, and the second one was a collaboration between Ron Germain and David Sachs. And they looked at neutrophil dynamics in the tissues of living mice. One was in toxoplasma-infected lymph nodes, and the second one was in leishmania-infected skins. And both studies showed what was termed swarm-like dynamics of neutrophils. And I guess you can see that here, that these neutrophils underwent phases of directed migration and then also clustering, a little bit reminiscent as of what some insect swarms are doing. And a little bit later, two other studies also came out and with again, remarkable results. And the one came from the lab of Paul Tukes, and he studied this massive recruitment of neutrophils in the liver when the liver is injured by a focal thermal liver injury. And also by Lai Guanyin and Wolfgang Weniger, who studied and used the sterile inflammation model where they had a laser-induced injury in the skin, where also neutrophils form something like swarm-like dynamics. And up to now, many more examples have been observed by many research groups, and we now have more than 30 inflammatory conditions ranging from sterile inflammation to infections with bacteria, parasites, and fungi where we see these neutrophil swarms with intravital microscopy. And we also start now to categorize these swarms. So you have here what we call is a persistent swarm, where a large number of neutrophils comes together to one focal spot. Here's the example of a sterile skin wound. Or on the right, what we call transient swarms, like here, where the swarms form clusters which come and go, yeah? Like here, for example, in an infected lymph node. And I guess the term swarming is justified, in particular when you look into parallels to insects. And here's also a beautiful YouTube video from National Geographic, because the response of the Asian honeybee is really pretty reminiscent of what we also have like with the neutrophils. So these honeybees like show a population response, which is very interesting. And here is an example where a hornet entered the beehive, and here if we place again, it grabs a bee and bites off the head. Yeah, and so in that minute, what happens is there's a population response of all the bees happening, and they form what is called a thermal ball. Yeah, they're actually beautiful studies, a little bit dated already. Yeah, about this response, and what is happening is that these bees in this ball, yeah, increase the temperature, and with that they also fry the hornet. So the hornet is killed in the end. Yeah. And so if we think now about our neutrophil swarms, also they have been shown to be protective. So definitely what they do is they contain a tissue damage from the healthy tissue. And what they also do is these clusters is that you have a concentration of microbicidal activity from a whole neutrophil population on one spot. On the other side, once these swarms and clusters form, they can also be destructive for the tissue, either by remodeling extracellular matrix, or there were also examples showing that host cells were cleared. So I then also started to establish a model for neutrophil swarming in the mouse ear skin, where we used the 2-photon laser to create small focal wounds, not bigger than 10 to 30 microns in the skin dermis. So we have one here in the middle. And then here in this 1.5-hour time lapse, you see how this happens in a tissue. We have in red some stromal elements, but you also see here in the middle neutrophils moving. And in green, you also have monocytes that are also recruited, but with a little bit of a delay. So first, the neutrophils form this impressive swarming response to this injury. And then when the neutrophils start to cease swarming, then you have these monocytes that also accumulate and start forming a ring around this neutrophil ball. Now, I mentioned before autonomous neutrophils. So it's clear that neutrophils do not follow preformed ECM paths in a tissue. But what made me now really wonder was like what makes them swarm. Yeah, if you look what makes them move that directly when they move and form these swarms. And another remarkable thing about neutrophils is that they have so many chemokine chemoattracting receptors on their surface. So they are really equipped with really a large number of receptors that makes them an ideal cell for tissue surveillance. And many G protein coupled receptors, GPCRs, means that they can also sense a lot of chemoattractants and chemokines, most of which can induce cytoskeletal changes and direct neutrophils in the tissue. So to understand which factors and mediators are now important for the swarming response, I started to set up an experimental system for screening mediators of neutrophil swarming, which worked in that way that we isolated bone marrow neutrophils from control mice or any kind of gene deficient mice. We differentially dye-labeled them, took them one-to-one, injected them into the ear, and then performed imaging. And the experiments looked like this, that you had control and knockout cells side by side, and you could see how they are moving. You can observe them during the different phases of swarming, even at the end, when this neutrophil ball starts to rearrange the collagenous fiber structure in the dermis. And then it was just fantastic to be at the NIH, to interact with many colleagues at different institutes that were very willing to share their knockout mice. And we could really do a nice screen here with primary neutrophils, and we focused on chemoattractant and chemokine signaling, also the role of adhesion and cytoskeleton. And I would like to highlight two findings. The first one, again, deals with integrin receptors. So the most important one is, I guess, this one, where we had now control cells as well as these tailin-1 knockout neutrophils. Again, as a reminder, these were the neutrophils that were deficient in integrin activation. Yeah, so they had the integrins, but in low affinity conformation. And as in the collagen gels, the recruitment of these cells, here these are the green ones, was totally fine. But you can already see here that in the middle of the swarm where the ball is forming, these cells had a deficit. And when we look here a little bit closer, you could see that these knockout cells were totally fine when they moved in the fibrillar interstitium, but then they exactly stopped there where there was the transition zone into the cell ball where only the wild type cells entered, which was a region that actually formed because the ball, this neutrophil ball, started to displace the collagen fibers, as you can see here, to the outside. So what happens is when the neutrophil cluster is forming, it rearranges the fibrillar extracellular matrix. It forms like a collagen-free wound zone where wild-type neutrophils can still move into, while the knockout cells could not. And here you see now both wild-type cells in red and green, just to visualize how they move in this collagen-free wound center on top of each other and continue to be motile. While on the next slide, I show you the integrin deficient. In that case, these were the integrin beta-2 deficient neutrophils. Here is this border and transition zone. They tried to go there, so obviously they still sensed some attractants, but they just could not. They slipped at this border and could not enter the ball of wild-type neutrophils. The second finding was an important role for the inflammatory lipid leukotriene B4, short LTB4, which mainly acts through its high affinity receptor LTB4R1, which is also called BNT1. And the phenotype here was also one during clustering. So LTB4 controls neutrophil clustering. And you see that here, when we had these receptor knockouts, and these clusters formed, the red control cells pushed out over time these knockout cells. And then there was a second phenotype, and this was that also LTB4 controls the long-range recruitment. So on the left side, here in yellow, you see the tracks of wild-type cells, and they are recruited from distant regions, while these knockout neutrophils, only the ones that were in close vicinity to the damage, yeah, they could be recruited, but the ones that were sitting far apart could not. And what we could also show was not only in the sterile tissue skin wound, but also in a setting where we had like a bacteria-induced neutrophil swarming and transient swarming. Also here, LTB4 played a role, which you can see here. So on the left, this is the video that you also already saw in the beginning. In a wild-type situation, in this bacteria-infected lymph node, you had these clusters that come and go, pretty big ones. And in a situation where we had the receptor knockout with the lyse and GFP to visualize neutrophils, the maximum that we saw was like these little neutrophils that formed these clusters. So with all these models, what we could show was in the end that it was even the neutrophils that were secreting the LTBF. So there was indeed intercellular communication among the neutrophils via LTB4 happening. That's important. So in an autocrine and paracrine manner, these cells can communicate with each other via LTB4. It's even a bit more complicated. I showed you that the integrins were not required for the recruitment to the cluster, but once they are in the cluster, also MEC1 and LFA1, both of these, both of them actually were important. And then also two other receptor systems, CXCR2 and FGFR2, are also kind of involved to a certain degree in this whole response. What I would like to mention also is that this LTB4-dependent neutrophil swarming is conserved across species. So our results were now all from the mice, where we showed that neutrophils release LTB4 and that it's also important together with CXCR2 for the clustering. There's beautiful work also done in zebrafish. Also, this swarming responses depend on LTB4. So this has been shown by the research groups of Renshaw and Elks, as well as the group of Milkasaris. And the Saris group had a beautiful paper in the zebrafish that showed that cell-cell contacts in these nascent clusters are important to trigger some calcium alarm signals. And this increase in calcium in these cells then actually triggers the LTB4 release. So these results are still pending confirmation in the mammalian system. And actually also LTB4 and CXCL2 are also important for human neutrophils. And I will show you later how one can study human neutrophils also in a dish and their swarming behavior. So what is now triggering LTB4? And these can be many factors. So many inflammatory triggers are known to induce LTB4 in neutrophils. This includes formyl peptides, which can come from damaged cells or also from microbes, wound signals, complement factor bacteria, zymosan. All of them in common is that they lead to a huge increase of calcium. This then activates a two-step enzymatic cascade, which then in the end leads to the release of LTB4. And this is what we work with. It's a multi-step model of neutrophil swarming where we have some first cells that can see damage in the tissue, like a sentinel cell. If cell death occurs, this can amplify the whole response, but it doesn't have to. So this is not essential for swarming, but it can act as an amplifier. And then I showed you that neutrophils themselves can secrete attractants and self-amplify their accumulation via LTB4, and also CXCL2 is involved. And then at one point, everything terminates and also resolves. So sometimes colleagues contact me and ask, yeah, we also see swarms and that's great, but somehow my swarms look different than your swarms. And then I say, well, yeah, this is very likely to happen because there are so many factors that can influence the swarm phenotype. And I would just like to quickly show what can all influence the exact swarm phenotype. So the first thing is, for example, do you have a tissue damage, a damage in the tissue, an injury, and how big is it? Because if it's rather small, it will only recruit fewer neutrophils. Then when it's bigger, of course, more neutrophils will be attracted. Then one thing will be also, do I only have like one injury or several injuries in the tissue, because they can start to be compete with each other. It will depend on the exact trigger factors that might trigger the calcium burst in the neutrophils. And then it also depends like on the location or the organ. And also if you have a lot of macrophages in your specific tissue or not, because it has been shown that macrophages, in particular for small lesions, like something like this here, they can be very fast and do a cloaking response and in a way enwrap this whole damaged tissue in a way that neutrophils become blind for it, and then they will also not swarm. Then also if pathogens are present or not. So some pathogens might be also more inert, but others maybe also cause something like a secondary cell death, which again could amplify the swarming response. And then also how many neutrophils are attracted, because the more neutrophils you have, the more secreted attractants from neutrophils you will have. And again, you will get this self-amplification. And also as a last point, do I have some other competing attractants, maybe from other cell types that are increasing something? So also this could redirect neutrophils, and maybe the swarming response will look different than in a different model. So and what we now worked on over the last years was the question, how does neutrophil swarming stop? And in our multi-step model, we are now here in this termination phase, and we asked about the stop mechanisms of this process. And we approached this very naively by saying, either is it something that the neutrophil controls itself, or is it something that is regulated by external signals? And we hypothesized that this is maybe something that the neutrophil can control itself. And we thought what maybe happens, so potentially through these accumulating neutrophils that are secreting attractants all the time, there might be high local concentrations of attractants somewhere. The reason why there were so many, a lot of may and potentially is we cannot really visualize chemoattractants and chemokines well in a tissue. Most of them are soluble and you will just wash them away. And what's even worse is we have no idea about local concentrations in the tissue. So these were all assumptions. But what happens at high local concentrations is a process called GPCR desensitization. And just here as a little reminder, immune cells can sense picomolar concentrations of a chemoattractant, and if it's distributed in a graded fashion, this is sufficient to induce chemotaxis. So a cell would not require all its receptor repertoire on the surface to induce chemotaxis. However, at high concentrations of an attractant, what happens is that usually all the receptors are engaged, or most of them, which can lead to the desensitization, sometimes also internalization of the receptors. And what can happen is that immune cells attenuate their responsiveness to this attractant. And an important family of proteins that's involved in GPCR desensitization are so-called G-protein coupled receptor kinases, short GRKs. What they can do is they phosphorylate the intracellular parts of activated GPCRs, which can lead to the receptor uncoupling from the G proteins, or sometimes, not always, internalization or sequestration of the receptors. Now, in immune cells, 4 GRKs can be expressed: GRK2, 3, 5, and 6. And so this is also the case in neutrophils. And what we did then was to systematically target either individual GRKs or even the whole family of GRKs by using and generating conditional knockouts for individual GRKs, or also using some already existing GRK constitutive knockouts. And in our studies for swarming, what the family member GRK2 turned out to be the most important one. And this already showed when we did our screening of these different GRK knockouts, when we looked at them side by side in an under-agarose chemotaxis assay, where we gave a gradient of our swarm attractants LTB4 and CXCL2. And in a wild-type situation, what happens is that cells move, but then at one point they will stop, because here they reach, obviously, the concentration where they desensitize. And the GRK2 knockouts, as you can see here, again, here the wild-type cells, they move and then they stop, but the GRK2 knockouts, they continue to move over that border, and even into the fields here where we had high concentrations of attractant. So here's again the quantification of that. The GRK2 knockout had a displacement almost twice as the control cells. Our quadruple knockout did not give any additional effect, and our other individual GRK knockouts just moved as the wild-type cells moved. And what was really remarkable is that this was not a general response like to all GPCR signals. So there were some chemotactic behavior to other GPCR stimuli like FPL1 and C5a, which acts through the C5a receptor, which there was no difference between the wild-type cells and the GRK2 knockouts. Our effect was mainly restricted to the GPCRs that require the neutrophil secreted attractant, so either LTB4 and CXCL2 together, but even the individual stimulation with LTB4 or CXCL2 was sufficient to see these effects. To really show that this is a desensitization effect, we took the gold standard assay to test it, which is looking at calcium fluxes and repeated stimulations with increasing concentrations of either LTB4 or CXCL2. And what you can see here is the response curve of wild-type cells, which will react, and you will have here calcium transients still with the first and second stimulation, but here it already is not responding anymore. And with the CXCL2 in this experiment, it's already here at the second stimulation that the cells are not responding. And if we now look at our knockouts, they continue to stay responsive. And this is also why they continue to move in these fields of high chemoattractant. So with this pretty cool finding and tool in hand now, we could ask now the question, how does this impaired neutrophil arrest that we see in these GRK2 knockouts impact swarming in physiological tissues? And we first looked at persistent swarms. And so here we again went to our sterile laser-induced tissue injury in the skin, where we had now wild-type cells in red, GRK2 knockouts in green. And as you will see, like the recruitment was both fine, yeah. So both cells in a way recruited the same. It was just in the end where then the green cells started to dominate the center and even push out the red cells. And this was something that was for us extremely interesting because this was the first knockout where a knockout did something better than the wild types. And this was like here, this improved clustering. Here's a smaller cluster, yeah, not big like the one here, maybe 25 cells or so. And also here, even in that cluster, you see that these knockouts, they will very quickly enter here the center and then take over and cluster around. And this effect was only seen here for the GRK2 knockouts in the tissue. Here a value of 1 means there's no difference between wild type cells and any of these other GRK2 knockouts. So it was only the GRK2. What we then also did was we teamed up with Eduardo Reategui and Daniel Iringer from Harvard Medical School, who are bioengineers and who had published a method to study neutrophils swarming in the dish. And they had described it for human neutrophils. So we also tried that out for mouse neutrophils, and we could also show that this is LTB4 and CXCL2 dependent. This works by micro-patterning, here, for example, Staph aureus bioparticles. So this is, for example, a stimulus that's known to induce LTB4. And what you just do is you add your neutrophils on top, and what happens is that then the cells start to swarm and form these clusters. And this is what we then also used with our wild type and GRK2 knockouts. And as in the tissue, again, now in red, our knockouts clustered more. Also, here's the quantification of this. So same thing as in vivo. And then we even moved to another granulocyte type, not neutrophils, but eosinophils, because also here it had been shown before that also eosinophils in an LTB4 dependent manner can have a swarming behavior in response to a number of worms, including C. elegans Dauer larvae, but had also been shown for schistosoma and some others. So we generated from a different mouse GRK2 knockout in eosinophils, and then did the same thing. So here's the Dauer larvae now, and you see this massive eosinophil swarm towards these larvae. And again, if we then quantified that, again, the clustering of these GRK2 knockouts was much better. What about the transient swarms? For that, we moved to a bacterial infection model, either Pseudomonas aeruginosa or Salmonella typhimurium, which we injected into the hind foot pad. The bacteria drain through the afferent lymphatics into the subcapsular sinus. There they induce death of the subcapsular sinus macrophages, which then leads to the recruitment of neutrophils. And then to see again wild type cells and knockout cells side by side, we did a 50-50 bone marrow chimera in a pretty complicated manner, where we then also had cells that were endogenous greenly fluorescent neutrophils and endogenous red fluorescent neutrophils. These were experiments that we did together with Sarah Eickhoff and Wolfgang Kastmüller, who also did the imaging for us. And when we then look here just at the static image, now we're looking into the subcapsular sinus of the lymph node, we again see that our knockouts now in green are in the center of the cluster, as we've seen before. And now when these transient clusters form, like here, for example, one, even then, again, even in the new cluster, these cells will cluster more. Yeah, so it's everywhere the same. So what I could show you, I guess, is that there is some form of self-control during neutrophil swarm termination. There's a self-limitation through GRK2 control GPCR desensitization during this phase of swarm aggregation. I showed this to you for sterile inflammation, infection for persistent swarms, for transient swarms, and there was also relevance for eosinophils. But there was even something more remarkable behind in these experiments. And this was when we then tracked the cells, because these knockout cells were not clustering better. They in addition were also faster in the tissue, and they also scanned more tissue regions. So they had larger track lengths, so they were faster and had this increased tissue scanning, which we thought this might be something very beneficial for neutrophils. Because the general idea was that the more neutrophils are scanning tissues, the more they will probably also sample bacteria and eliminate them. So it's the current thought. So we asked, is there any consequence for bacterial elimination? Maybe even something that we could use therapeutically. Yeah? So we took normal mice as well as then neutrophil-specific GLK2 knockouts, infected them with Pseudomonas aeruginosa, waited for 8 hours, and measured then the extracellular bacterial burden. And then, strikingly, our knockouts were actually worse in eliminating the bacteria, and they were worse in the clearance of the bacteria. And yeah, and so this was a little bit of a puzzle for us, because when we looked at things like neutrophil differentiation, bacterial phagocytosis, ROS production, MPO and neutrophil elastase release or net formation, these knockout cells were not impaired in these processes. So to understand now the why and how, we also came to the limits of our intravital microscopy. And what we started to do was we started to mimic the infected lymph node subcapsular sinus in the dish by bringing all its components together, meaning macrophages, neutrophils, as well as bacteria. And I will show you this step by step. So what we first did was we put macrophages with Pseudomonas together, in addition with a cell death dye, that we can see how bacteria are killing macrophages, which are now turning red here. Yeah. As a swarming person, I just got completely crazy, because here, as you can see, also the bacteria were swarming. I was like, oh my God, now we also bacteria are swarming. If you read a little bit carefully in the literature, this phenomenon had been described for Pseudomonas. It's called peck swarming. Yeah, it was interesting, but well, what you can nicely see that you have here this peck swarm, and then exactly there is also where the macrophages a little bit later will be dying. And then we added our neutrophils, and we were very happy to see that also then the neutrophils now here in blue also accumulated around these dying macrophages where we have the locally proliferating bacteria. And then we used the system and took either our wild type neutrophils and gave them 3 colors. So the macrophages and bacteria are still in that assay, but just unlabeled. Or we took the knockout cells and looked at these clusters. And then we looked at the dynamic behaviors between both of them. And while the wild type cells did something pretty controlled, a pretty controlled clustering, our knockouts behave in the lab, we call like a wild bee behavior. Yeah, they really, I mean, they were fast and clustered also more, but it was just very uncontrolled. And the consequence of that was that the containment of these locally proliferating bacteria was never that complete, like in the wild type situation. There were always some spaces where the bacteria could escape. And when we then also in these, when we also here then determined the extracellular bacteria, there were much more growing extracellular bacteria in these culture dishes. And this was the explanation for us, what we also saw in the tissue. So I'm coming almost to the end. So I hope that I could convince you that neutrophil swarms have and follow principles of self-organization. So in the initial phase, when neutrophils start to secrete swarm attractants in order to self-amplify their recruitment, and then also later, when the same attractants, once they accumulate, because you have this increasing number of neutrophils there, also self-limit and actually stop the cells, which is important for the containment of bacteria. So for the last 4 slides, I just prepared something which, because this usually comes up in question and answer sessions, and that's the question, how are neutrophils actually redirected between clusters? How does it work? And it comes back to this question, because now I talked all the time about the neutrophils are coming there, they desensitize to the swarm attractants, and this is GLK2 dependent, and then they stop there. But how does it work that they can move, especially in these transient swarms, now between the swarms? And I can show you, we did a proof of principle experiment here now in the skin tissue by using neutrophils that we could also photoactivate by using mice that had been described before from Michel Nussenzweig, photoactivated PA-GFP mice, which we crossed here with tomato. And what you can do is you can create a cluster, which we did, and then we photoactivated only neutrophils from that cluster. And then we did a second new laser-induced lesion. And you see that also neutrophils from other regions are now recruited here, but you can also recruit quite a number of neutrophils here from this established cluster here. And what we think is happening is the following, that this new insult what it is actually something where other attractants, which probably belong more to what we call end target attractants, I will come to that in a second, is released, which are GRK2 independent, and this is why they can be a new stimulus for cells that were desensitized here to the swarm attractants. And with that, neutrophils are now pretty flexible in the tissue. Yeah, so they know they can self-amplify, they can self-limit, but they stay reactive to new insults in the tissue, where then, once they accumulate, they can still undergo the self-amplification and self-limitation process. And all that is also in agreement with a concept that has been very long in the field, and this is that what we call end target attractants, like for example C5a, like this complement factor, or formule peptides like FMLP, which could be derived from microbes or dying cells, can override intermediary attractants, which we consider these are attractants that are secreted from cells, like for example CXCL2 or LTB4. And this comes back to beautiful papers from Ellen Foxman and Eugene Butcher, as well as Brian Hyde and Paul Cubes. So our study is not at all contradicting this concept. I even think that both aspects complement each other. And with that, I come to the end and would like to acknowledge my crew of very motivated students, both the current lab members as well as former lab members. So the GRK2 neutrophil study was driven by Korbinian Kienle, a PhD student, and finished by Michael Milan, Katharina Glase, and Maximilian Epple in the lab. The study on macrophages that was just recently published was done by Neil Patterson, also a PhD student in the lab. And we had many very helpful collaborators during the GRK2 study in neutrophil swarms. And then, of course, funding agencies and also the Max Planck Institute of Immunobiology and Epigenetics, which very sadly will soon be turned into a Max Planck Institute of Epigenetics only. And yeah, let's see where the immunobiology will be moving within the Max Planck Society. And with that, I'm finishing here, and yeah, and I'm looking forward to all the questions on Twitter.
Terrific, Tim. That was wonderful. Really, picture speaks 1,000 words. Those are really beautiful images. Okay, so just to remind everybody, we do the questions here on Twitter, and you can reach Tim for questions on this really interesting work at Lammermann Lab. Thanks for joining this week's meeting, and again, just a reminder, next week's speaker is Yannick Crow. So we'll see you all next time. Thanks a lot. Thanks a lot, Tim.
Bye.
Appreciate you doing this. Yep.